Welcome to the Data Science Institute's virtual seminar series. My name is Dr. Sarah Mackey from Lawrence Livermore National Laboratory. Here at Lawrence Livermore, data science has become an essential discipline in many of our key program areas. LLNL is home to many challenging datasets, as well as home to some of the world's most advanced supercomputers. Our data science staff work in a variety of areas, including machine learning, artificial intelligence, big data analytics, statistical inference, predictive modeling, and uncertainty quantification. The Data Science Institute acts as a central hub for our lab's data science activities. We host events like this seminar series in order to introduce new ideas and potential collaborations to our laboratory staff. We invite speakers from outside the laboratory, from the Bay Area and beyond, to share their innovative approaches with our data science community. We're pleased now to include a wider audience in our seminar series through these recordings. You can read about past speakers at our website at data-science.llnl.gov, or you can email us at datascience@llnl.gov. Thank you and enjoy the seminar series.
Now ready to introduce our speaker, Anna Kenny. Dr. Kenny is a postdoctoral researcher in the Department of Statistics at UC Berkeley, working primarily at the interface of computational statistics, machine learning, and optimization applied in the domain of biomedical sciences. In fall of 2021, she earned her PhD in statistics and operation research at Penn State, and there she was a Sloan Scholar and she was a biomedical big data to knowledge fellow. So I've invited Anna today to discuss her work on simultaneous feature selection and outlier detection. Anna, when you are ready, go ahead and take it away.
Thank you, Sarah, and thank you again for the invitation for everyone attending today. I'm now at UC Berkeley, and this is work, I guess, mostly done the last few years during my PhD at Penn State with Luca and Solia, Francesca Chiaramonte, and Giovanni Felici, with some newer work that has happened in between. So I like to try to ground everything in an underlying problem that heavily influenced and I would say kind of inspired a lot of method development in this project and throughout PhD in general. And so that is the intervention nurses starts infants growing and healthy trajectories, the INSIGHT study, headed by Professor Ian Paul of the Department of Pediatrics and Public Health Sciences, along with several colleagues at Penn State Hershey Medical Campus. And so in general, this is a longitudinal study that began with around 200 families who were enrolled with the birth of their first child. And then as time goes on, we're also getting additional information on any siblings that might have been born into these families. And so these families have been great. They come in, everyone's getting poked and prodded, and various types of data is being collected, say at the blood, feces, and saliva samples to generate what we're referring to as omics data. It's genomics, metabolomics, and so on. In addition to sort of all of these samples, we also get a lot of survey information, the type of diet they might have had the kids on as time goes on, which, you know, we trust people are being honest as they're supplying this information. And so with sort of all these different facets, as we're trying to study sort of child growth and development, this called for a really interdisciplinary group of people to try to tackle different corners of this. And so we have professors and postdocs and research associates from areas of genomics, bioinformatics, computer science, statistics, is where I came in, all working together on different corners of this overall project and initiative. And so as I mentioned, this is a longitudinal study. And so just to give you sort of a snapshot, each curve corresponds to a given child in the study. And so we can see how they're growing over time. I think by the time I finished my PhD, the kids were like 4 years old. And so they are, I can gauge how old I am now based off as new information comes in with these kids. And so there's sort of different rates at which they're growing. Here we're going to focus more on some summary measurements of their growth in this microbiome case study. But we have other work that really try to exploit and use these full trajectories and slopes So if you're interested in any of that corner, I'm happy to talk more offline on that component as well. But so as I mentioned, this is a pretty multi-omic approach. So we have genetics data, microbiome, epigenetics, and metabolomics that could all contribute to other risk factors of, say, early weight gain or childhood obesity. I was fortunate to work on sort of these two main corners, and will probably only have time to really touch on some results from the microbiome side, which is also a corner that there was sort of this initial hypothesis that there were some extreme values going on with a few of the bacterial abundances. That is to say, I guess everything isn't limited to this one main study. So we also had some more recent work that came out using some extensions of the methods I'll go in today on investigating drivers of honeybee loss. We got a nice cover with the special issue in Robust Statistics that came out with a bee in the background. And that was with Luca Insolia, Martina Calovi, and Francesca Chiaramonte. And so this dives into more of a classification problem and other ways you can sort of use the flexible framework we're proposing here into all these different problem domains and make it pretty specific to the type of problem you're looking at. And so I think trying to keep things very applicable and generalizable is sort of an emphasis on this work. But I should mention that while we do work on some modeling approaches, especially now at Berkeley, that are pretty, say, prediction-centric, in this particular case, especially in the INSIGHT study, we're working with clinicians, pediatricians, people who want essentially a set of features or something to look into, right? So like, what genes should we take a closer look at that might be related to weight gain? Which bacterial abundances do seem important? And so that's what framed a lot of these questions around performing feature selection on these pretty large, complex data sets. And so some of the challenges I wrote down here that went into different projects, and they're probably, I would think, challenges that a lot of us have encountered as we're working on real data, especially real complex data, even outside of biomedical sciences. But so, for instance, we have severe undersampling, meaning we only have around 200, say, individuals, children in the study, with, for instance, in the genomics case, we have like 1 million potential genetic variants that could be contributing, right? Interdependence or collinearity among the features or units is an ongoing issue. It's very difficult to decipher where the real causal factor is when you have all these relationships going on across your features. The microbiome data in particular, you have things that are coming from the same phylum, for instance, and so you have correlations going on there. I guess with things like growth or complex diseases like obesity, you also have this issue of weakened or uneven signals. So we're not expecting very clear, obvious, here's the gene that fixes it, that addresses everything, but maybe weaker signals coming from a collection of different features and components, right? Which adds to the difficulty. Finally, we won't go on to this one as much, but we do have fairly complex responses that are other options to use, such as the more longitudinal curves in place. And then getting into, especially in this project, was the potential of unit contamination. So I will stress that the INSIGHT study in particular was very carefully done. The nurses were trained in all the measurements they were taken. When you compare, let's say, electronic health record data or some of these big meta-analyses going on, there's a big difference in like the measurement noise level you might find in what we have in-house versus what we have in some of these other public data sets. And another benefit is because everything's done in-house, it's easier to sort of check with like, oh, I see something strange. What does that correspond to? And you have everyone who was part of the measurement process sort of involved. But even with all these safety checks, there's still the chance that something goes wrong. There's some weird contamination present. If not contamination, maybe there's some subgroups within your population you weren't aware of, and they're skewing your results. And so outlier detection kind of became an important aspect. Finally, a lot of the data we have is pretty sensitive, especially, say, the genetic side. It's all coming from children who live in the Penn State Hershey area. And so we did some work on how to release results in a differentially private way. And so again, today is going to be more focused on challenges 1, 2, 3, and 5, with, I guess, more emphasis on this paper highlighted in orange. For those who want more details on it, that's up online. But we kind of have sequentially built on different work to get to this point. And for those maybe familiar with functional data analysis, we have some extensions going into working in that framework. Again, happy to discuss offline. But okay, very simply, here's the type of setting we're considering when I'm talking about feature selection and outlier detection. So let's start very simple. Say we're looking at the correlation or the relationship between a given X and Y. And so at least with the way I've laid this out, it's very clear there's kind of this two clustering effect. The majority of your data is sort of in one area versus you have this shifted subset of observations in another portion, right? And so this is coming from what's often used to represent outliers, the mean shift outlier model, where a subset of your points, if you think of your typical regression setting, these are now, their residuals are coming from a distribution with a shifted mean, and each from a potentially different mean. So they can all be shifted in other directions. And we assume that this shift can be occurring both at different x's as well, so across different features and on your y's. And so if you didn't see this, or for instance, you had more than one X value, so it's really difficult to clearly detect that this effect is taking place, you could easily just quickly run through some correlations and say, okay, I'm not seeing much of an effect, versus if you actually try to look at the core of what most of your data was trying to indicate, you would get a very different effect in place, right? And so that's one of the issues with these things like outliers, influential points, leverage points. So this idea of just ignoring part of it or trimming and focusing on this core aspect is certainly not new. These are considered more robust fits using trimmed loss versus, say, your standard ordinary least squares regression. Dating back like to Rousseau in the 80s, where he proposed least median squares regression. The idea when you write it out is pretty simple. Instead of minimizing over all of your samples, you're now going to trim out a set of Kn observations that you consider to be outliers, and these turn out to be those with the most extreme residuals. So you're actually minimizing over these order statistics, right? And so the trick here is you need to find out which of these are the most extreme residuals so that you can trim them and refit your model. But you run into a problem, and this is why we argue you need to do this simultaneously. Because if you have outliers in your model, as you do in this case, that's going to impact which features seem to be important. So if you're trying to do any sort of model selection, your outliers are, of course, going to change which features at the end you think you should include in your model. Conversely, the way we're trimming, it is based off of some underlying assumed model. So the features you have in here are also going to affect which of the observations you end up trimming. So contamination affects your features, but the features you started with, if you have a misspecified model, are going to affect what looks or doesn't look like an outlier. And so trying to do a simple two-step procedure of like cleaning first to get rid of outliers and then doing model selection doesn't quite work. And so we then thought, okay, how do I do this more simultaneous approach instead of trying to tackle one problem or another separately? That's where we get into it. It's hard to say, but SFSOD Framework, Simultaneous Feature Selection Outlier Detection, where we're still trying to keep our trimmed loss, but now we're going to directly constrain the number of features that we're going to allow in our problem. And so this is saying I want to minimize my trimmed loss, but I'm only going to allow KP features into my model at most. And directly up here, we're also constraining the number of outliers. Sorry. the number of outliers in our problem. And so what we've done here is we've formulated a best subset selection. So this is considered a hard penalization approach. And that's because I'm directly saying I want to find this best combination of KP features that will still minimize this best combination of observations where I'm trimming out KN of them. So I'm not only looking at different groups of features, but I'm also now trying out different samples simultaneously to minimize this loss. And this is in contrast to what we consider soft penalization methods, such as MM and S estimators. For instance, they have like sparse least trim squares based on lasso regression. And that's because instead of solving this problem directly, that they really rely on heuristics. And many of them also rely on, say, a somewhat clean initial sample of data to work off of, and you do this iterative subsampling scheme. So big benefit is that they tend to be very fast, quick to use. They're not really solving this combinatorial problem directly, but maybe they might estimate it. For those that I know some people don't care as much on the theoretical side, but there's also, it's harder to develop a stronger theory when you are working in this heuristic setting as well. Or at least they require more assumptions than what we'll require under this combinatorial setting. So our proposal in general is going to have stronger theoretical guarantees under fewer or weaker assumptions, which for me, especially in feature selection literature, the assumptions needed tend to be unrealistic. So if we can pull that back in any way, that's already a big plus side. But we're also retaining more interpretation taking this approach. So another thing about soft penalization is you're not directly calling something an outlier. You're more just weighing the different samples to try to figure out what might work best. So this one's more direct. You're directly selecting features and trying to detect what looks like an outlier. And once I do that, can I see, does that make sense in my data set? Is there a reason this seems to be contaminated? And then finally, because of the way we can reformulate this problem, we get some nice optimality guarantees from the algorithmic point of view. So I can guarantee I'm within 5% of actually solving the best combination of features and samples for this problem. Okay, but there might be the question of why am I doing best subset selection, right? For those familiar in the feature selection literature, I would say regularization approaches are a lot more common. So thinking back to this general framework where you're minimizing some sort of error measurement and subject to a penalization term. And so this penalty, there's a lot of work and stats on different types of penalties one can use. There's lassos where you do an L1 here so that you get sparsity in your shrinkage. The ridge is very common, might help more, especially under noisier regimes. Elastic net's a combination of the two. SCAD penalty. There's a never-ending supply of different ways to penalize here, right? And so what we did was we kind of went back to one of the first initial approaches one would have tried with feature selection, which is the zero norm. Not quite a norm, but what it does is you're just doing a direct bound on the number of non-zero coefficients rather than the size. So we're avoiding any bias that's induced that you would get from, say, a lasso because of the weights and the way you shrink your regression coefficients. But instead, you're directly just saying, you're either in or out of the model, and I'm only allowing K of U in the model. And so best subset selection, this combination approach, while being one of the first things you would try, was also for a long time thought of as simply not feasible for more realistic problem sizes. And even textbooks in 2013 were saying, okay, yeah, it's great, you can do it, but only if P is less than 40 features, which most problems are going to have a lot more than 40 features. But there was sort of this pretty foundational paper that came out in 2016 that showed actually, if we use some modern optimization approaches, because the optimization community has been actively working on this type of problem for like the past 10 years or so, we can now solve P in the thousands in a matter of minutes, again, using mixed integer programming to do so. And so this big gap started, I would say, almost a huge debate in the stat community over using the L0 norm versus your usual lasso. But now it finally became a competitor for the first time in quite a while. And so part of some of the benefits, I would say, that go into doing this direct best subset selection is not only sort of the intuition behind it, but you also get some nice theoretical guarantees. So I haven't specifically said what type of loss function we're working with here, but generally one can prove that you get a pretty high breakdown point. In fact, the maximum breakdown point possible. And for those unfamiliar, breakdown point is sort of a common concept in robust stats literature. And so this is the largest proportion of outliers that an estimator can tolerate before it becomes arbitrarily biased. And so as long as our trimming level, that's our KN, so the number of outliers we're trying to trim out, is at least as high as the number of samples present, or the number of true outliers present, you're going to reach this best breakdown point. So what that means is, as long as I'm trimming more than I should, I'm okay. But the breakdown point itself, it really only protects us against the worst case scenario. So what I'm saying here is, as long as my trimming level is high enough, my estimator resulting from this procedure will not have infinite bias. It'll have a finite bias. So all I'm guaranteeing is we have finite bias at the end, or the maximum breakdown point possible for finite bias. So that is also true for other soft penalization methods like the least trim squares with lasso, estimators, least quantile regression, all have this high breakdown point property. And so we wanted to kind of do more than just saying, hey, we don't have infinite bias. And so when you get into a more specific loss, so L2 loss, thinking of like mean squared error, this is where we're able to prove what we're calling a robustly strong oracle property. And so in feature selection literature, we have the oracle property, which says that with probability tending to 1, you are going to get your true set of features. You are going to eventually pick them up. Again, this is all asymptotically, right? And so with the robustly strong oracle property, what we're saying is, even in the presence of outliers, we're still going to pick up this true set of features. And in fact, I can guarantee that not only will I select the true set of features, but I will detect the true set of outliers. So I'm able to establish both theoretically. Oh, and I should also mention that, again, this is all asymptotics, and so finite sample theory gets tricky. But here we can also allow the number of features to grow exponentially with the sample size. And so all of the results I'm showing are going to hold in this like ultra high dimensional setting I mentioned earlier as one of the main challenges. So at least theoretically, this should still work, even under really high dimensionality. The last property we have here is optimal parameter estimation. And so this says that we actually achieve optimal prediction error as well as risk minimax parameter estimation. So our best solution is actually the best under the worst case scenario as well. So we're not only getting nice selection consistency, but we also get good parameter estimation as a result of this, which the parameter estimation kind of comes naturally because remember, we're not inducing any bias. So we're looking at kind of these OLS estimators in this setting. I think I saw a question flash, but I don't see it now.
Yeah, we got it. Sorry, I missed it. A question from Paul Munger asks, does the robustly strong oracle property only hold if outliers are independent in some appropriate sense, say from inliers?
I see. So like your samples corresponding to outliers, like two siblings, for instance, that are dependent on one another.
That
One
kind of
shouldn't have
thing.
outliers.
Yeah, yeah. Like I'm sort of imagining, what if your outliers are allowed to try and masquerade as inliers by being correlated with certain inliers or something like that?
Gotcha, gotcha. Let me think. So, yes, I want to say I don't think there's an explicit assumption on independence among samples in that way. Uh, but I do wonder if it sort of arises from the asymptotics here. Um, so I'm not quite sure, I guess is the answer, but I think you're right that there is some issue here when you have the dependence among the observations as well. I think especially in the optimal parameter estimation, since we are leaning heavily on some like OLS assumptions to work with that one. Yep.
Yeah, that's interesting. Maybe I'll just have to pay very close attention to the following slides so that I can try and think about it.
Well, no problem. Well, time is actually a little tight, so I won't get to go into like the proof details, but I'm also happy to discuss more later. But yeah, okay. Very great question. Thanks. So as I mentioned, unfortunately, I won't get to dig into each of these conditions, and I wasn't sure what type of audience I'd be with, or that would be super interesting. But also happy to discuss with anyone who cares to look. But with these theoretical guarantees, I guess I also like to always put out the caveat, and even as Paul pointed out, there are other assumptions that will probably break this down, is that this is all asymptotic in nature, and while the underlying assumptions aren't as strong as the ones needed for some of these soft penalization methods, it's still theory, and it's still going to be very different than what we see in practice, especially when we're dealing with really noisy regimes, right? And so hopefully the numerical results might help complement this as I present it to you. But that gets into the question of how in the world do I solve this thing, right? It seems great theoretically, it might work okay, but I'm still talking about a pretty heavy combinatorics issue, right? And so recall this is our setting where we're minimizing, again, this trimmed loss, which we can rewrite in terms of our order statistics, subject to some penalization on my features. Now, there's this nice property you can prove where if you introduce a new set of features phi corresponding to samples, you can actually rewrite your objective with an additional constraint as this minimization problem. And I'm going to break this down. So for instance, my betas correspond to my feature selection, so that's fine. So if the beta 1 is 0, that means that X is not being used in my model. And so what my phis are going to do is they're going to help zero out the residuals corresponding to the samples that I don't want anymore. So a non-zero phi, a phi of 2, let's say, is going to take on the value of that y minus x beta, and that's going to cancel this guy out. And so I've effectively removed that sample from the problem. And this is, and by doing it this way, so my phi is a vector of size n, the number of samples. But what I've done is I've reformulated the problem as a feature selection problem on not only my betas, but now my phis. And this isn't a new property, by the way. I'm not claiming this. This has been established, but not really used because of this reliance on like your L0 selection. And so with this, I can now do feature selection on my betas, and then picking out which phis are going to be non-zero, I can also directly be removing or including certain samples from my fit. Now, in order to completely do this problem, you have to add on a series of more constraints, which let me break down a couple of them for us here. And so the big trick, I would say, to solve this problem is introducing these integer or binary variables, which act as on and off switches for their corresponding features. So the z betas go to beta, the z phis go to phi. And so what this is saying is if Z beta 1 is going to be non-zero, or it's going to be 1, then that means beta is included in my model. Same thing with the phi's. And so this is all done through the use of big M constraints, for those that might be familiar. So again, if the Z's take on zeros, then my beta will be 0. And so these Z's are actually not only kind of a dummy variable to help us do this direct selection, but they actually give us some pretty nice tools to play around with flexibility, like different types of problem structures. So one can imagine that once you've introduced these Z's corresponding to each feature, right, you can easily incorporate things like if-else statements. If this feature is selected, then also select this feature, or don't select this feature, right? So if there's some sort of domain knowledge that comes with certain types of features, it's much easier to incorporate that in this type of framework than it would be in some of your typical feature selection frameworks. So even separate from the outlier detection, I would say in general, this is one of the big plus sides of mixed integer programming, is the fact that you can really play around with all of these integer variables. And so just some pros and cons. So as I mentioned, we get this nice flexible framework. We can directly control the level of sparsity in our features, along with the amount of trimming we want to remove from our problem. We have all these computational advances, heavily leaning on the optimization community for all the work they put into it. And with that comes these nice optimality guarantees. Because the way this works is you end up solving for some feasible upper bound solutions, you get provable lower bounds, and you keep going until you can converge, and you're within some, like say 5% of the truly optimal solution. Now, I'm not gonna say this is, we're at the point where this is faster than doing a lasso or doing these other convex methods, because this is very non-convex. It's an NP-hard problem. It's still a very difficult problem. You do need to rely on these sophisticated algorithms to run it, which means if something goes wrong, it might be harder to find out what's going wrong. When I say sophisticated, I'm thinking of branch and bound algorithms with combinations of cuts. There's just a lot going into it now because these algorithms have been so finely tuned, I would say the last 10, 15 years. And so at the end of the day, they're going to still be more expensive than maybe your typical regularization approach, right? The P in the thousands is still going to take a few minutes to run. And if you're trying to run that many, many times over different data sets, things can easily explode. And we're certainly not at a point where I could shove my genetic data into here with millions of genetic variants and get anywhere near optimality in a realistic amount of time. But there has been a lot of work, pretty specifically in the feature selection literature, dealing with optimality and proposing some other heuristics to try and speed things up. We have a paper at JCGS that goes through just kind of tips and tricks to go about speeding this up again in the feature selection framework. The downside with the outlier aspect is it does make the problem even trickier. So one example is often the solution we're trying to reach, we may have already gotten there, right? But we can't guarantee optimality yet because we have a very weak relaxation. So our lower bounds take a very long time to get up to the point where we can say, oh, for sure, I'm within 5% of the optimal solution here. And so that's what really takes up this computing time, is often just trying to verify this. So one option is to just do an early stop. I don't care, I think I'm good enough, let's go with that solution. But you might not necessarily trust the solution you've gotten if you take that route. Another common approach is to use perspective cuts, which can help strengthen the relaxation. And so there's a series of papers, if you're interested, by Frangioni and Gentile. Again, the optimization crowd has been doing this a long time, so all these papers are back in 06, 09, but they're now sort of being seen again. And the main issue here comes with the introduction of our phi terms, because if you write out this quadratic form, you're no longer getting this nice diagonally dominant component. Main point being perspective cuts are no longer effective. So one of the main tricks we used to use to help improve the speed of the lower bounds just fails in the outlier detection framework. If you're just doing feature selection, it's great and also helps speed things up there. So we have some ongoing work on trying to extend this to the outlier setting. So that is to say, there's a lot of open areas, I guess, to poke around with if this interests you. Okay. Now, I also wanted to touch on some of the flexibility that I mentioned. And so different ways, depending on the problems you're working on, if this is interesting, you might be able to adjust and play with it, right? And so one being, you can easily include feature or observation groups. So if you think of things like the group lasso, this is a very easy extension. So at least in genetics, we oftentimes may have blocks of features that are highly correlated due to their linkage disequilibrium. And so with high correlations, they're usually acting as proxies for one another. So in practice, we say if you picked one of them, it's as good as picking the rest of them, right? And so in our feature selection, outlier detection algorithm, it might be nice to also say, okay, if I pick one of these guys, I want all of them, or I do none at all. Another common application here would be if you're trying to include main effects and interaction terms. So if you're trying to do selection across those, if you include an interaction, oftentimes you also want to include a main effect. So you're just pairing up or grouping different variables that you want to be selected together. Again, those who might be more in the functional data or longitudinal crowd, or even if you just want to include, say, spline bases functions, and so maybe make this loss more flexible. It's easy to also include group constraints on the set of bases coefficients corresponding to a given spline representation. So again, I just want to stress it. Flexibility is really nice here if you have some of the time to let it go and finish computing, right? Another one, and this actually helps speed some stuff up, is cross-validation. So because we now have these new set of Z's corresponding to my samples, I can very easily include or exclude a fold of data from my problem. So you're not necessarily completely resolving each time, but you can adjust your constraints and do cross-validation using this technique as well. The last one I wanted to touch on is penalized logistic regression, in part because it's not quite as direct as the other couple examples I showed. And this is also the extension we ended up using for the case study on the bees I showed earlier. And so here we now have to reformulate our objective and add on some constraints using exponential cones to minimize the deviance for logistic regression. Main point being, you can even add conic constraints now to mixed integer programs. So there's just a lot of flexibility here. Now, the mixed integer conic program is going to be even more difficult than your baseline case, but it is an option. The software is freely available. We have all of our stuff up on GitHub, coded up in Julia, and using solvers like Gurobi, which has free academic licenses. So again, I would say it's pretty accessible for those who are interested in trying some of these things out. Okay, if I didn't miss any questions, then I'm going to get into numerical results. It's good? Okay. So I hopefully have made some case from an intuitive standpoint of why you might try this. But let's get into simulations. And so just right off the bat, for the simulations, we are contaminating not only your response, but also features involved in the underlying model. We're assuming a sparse scenario. So say only 5 out of 100 features are going to be truly important for a given response. And so the goal here is to find the 5, as well as trim out the outliers, right? And so for our comparisons, we do one against the Oracle solution. So this is if you fit an OLS with the correct set of features and completely removed the outliers, what's your prediction error looking like? How does everything compare? And then we also have two other, these are again soft penalization methods, the sparse least trim squares, which you can think of like a lasso here, as well as eNet LTS, which is an elastic net approach to this. Again, these two methods also have very nice R packages available, so easy to use and do your own comparisons as well. And so just to summarize before I throw a bunch of plots at everyone, but what we found was we did get competitive prediction error. So the root mean squared prediction error converges faster to the Oracle for us than others. And by converge, I mean we can hit a similar amount of accuracy with fewer samples. So we need less power in order to achieve that level of accuracy. And here's where the main difference really happens, I would say, between us and these softer approaches, is that ours tend to be sparser and more interpretable, and others tend to have higher false positive rates. So what this comes down to is sort of a really dense model versus a sparse model. And this isn't just the case here. The work we did on strictly the feature selection framework, again, using these L0 norms and mixed integer programming, also showed that they were much sparser than the other regularization methods. And so this tends to be kind of the issue with like, even the lasso, other people have proposed using it more as a screening tool versus exact selection because it has this tendency to over-select in practice. But the benefit of over-selecting is it's more likely that you at least collected everything you needed to, right? You might just have too much now. And so the downside to some of the L0 implementations is that you might have a higher false negative rate, which we did see here under lower sample sizes, but the overall false negative rate was very low in general. It wasn't like a 20% versus a 5%. Everyone stayed pretty low. But I would say even outside of outlier detection, when you are maybe categorizing these methods in your head, the L0, I would say in general, tends to be pretty sparse, very interpretable, maybe potentially too sparse would be the downside here. So in terms of competitive prediction error, this is one simulation we did. Here we have 50 features versus 200 involved. The x-axis is the growing sample size. So ideally you want to see prediction error dropping as you increase sample size. And this is average error over 100 different reps going on. And so in gray, we have the Oracle, and in orange is us, and then we have the two competitor methods above. And so by far, you could see that, again, we are getting very close to the Oracle with fewer samples needed. I would say this is the big one, though, for the sparse interpretable solutions. The other two methods, you're looking at 60% false positive rates, 20%, versus we basically at 0 across the board, maybe including a couple false positives here and there. But in general, very sparse, but we're still hitting the correct features even though we're so sparse. And this is also true when we're doing this detection on the PHIs. So we're also picking up the correct set of outliers in our fits. Conversely, we do get higher false negative rates under the low sample size. So you can see here, we're above the other guys. But again, given the scale, we're not doing that much worse. And in general, most of the methods aren't doing too bad for the false negative rate. I'm actually not sure why the two methods kind of increase so much under higher sample size here. Not the case under PHI. And so in general for the simulations, I would say it's about what we'd expected given the theory as well as previous results for feature selection. So the biggest thing would be maybe be careful with how sparse things are getting. And so while that sort of complements the theory, I'm always doubtful of simulations, even though we add more noise to the problem. We added We didn't go as extreme with outliers to see how much results were changing, and we still had pretty consistent results. It's still very different once you dig into a real data set, and especially one as challenging as insight, where we don't have many samples to work with. And so now, I think with the last 10 minutes, I'm just going to get into our case study if there's still no questions. So here we're looking at the microbiome, and this is building off work that also came from other people in this overall group that was published in scientific reports, where the questions addressed in their work was, is there a relationship between the oral microbiota of infants and mothers, as well as early growth of the children? And so early growth here, again, we're not considering the growth curves quite yet. That's ongoing work. But in this instance, we're looking at a measurement called conditional weight gain score, CWG. And so this is common in like pediatric literature. It's essentially a score showing how extreme weight gain or loss was in the first 6 months or so of life for these infants. And with Insight, we actually have a lot of measurements between 0 and 6 months, as they came in pretty regularly to fill out these surveys and everything. And so the other question was, if there is a relationship, what specific bacteria seem to affect these patterns, and does it tie into something? Does it make sense? And just to reiterate, here and in most omics work, these relationships are going to be pretty complex and subtle. Again, we're not expecting a huge loud signal to indicate, hey, this is what's causing a child's weight to suddenly drop, right? In addition, we have pretty limited sample size, which only adds to problem difficulty. Our questions are building off of this, thinking, can we find, or do we find similar relationships when we try and trim out potential outliers? If we do trim anything out, does it make sense? Do they look like outliers? Is there other information we could find on the children as to why they might have more extreme values? Is there a practical relevance to what we're finding, essentially? And then does our approach outperform competing robust methods? And we tried some non-robust ones in terms of prediction error. So, well, let's hope we at least are predicting similarly still with other approaches. And so again, here we compared against a non-robust approach, just a simple lasso to see how well it did. Along with sparse LTS and ENA LTS, we don't have any underlying truth to try to see if we're actually doing this correctly. We can only really go by current literature to see if our results make sense, as well as prediction error. And so again, we did see this sort of discrepancy between our approach and the existing methods where we had sparser solutions. The others were unfortunately pretty dense, talking 50, 60 variables being included, very difficult to interpret or to kind of return back to the clinicians and say, hey, you should take a look at this type of abundancy, right? Which, keep in mind, is sort of the underlying goal here, is to go back to the experts with some recommendation for them to look into. We also had competitive prediction error with lower or at least similar trimmed prediction error in the test splits. And I mention this because we tried to do this training test comparison multiple times so we get some stability versus trusting a given split. But under the assumption that there might be contamination in the data, that means even your test samples could potentially have contamination. And so we looked at trimmed error just to be safe. Finally, we followed a lot of the pre-processing that was done in the original paper, where because of the nature of microbiome sort of data collection, there's going to be a lot of zeros initially because some children don't have any measurements for certain abundancies. And so in general, they were aggregated based off of actual biological structure, so belonging to the same phylum, grouped together, and some correlation-based grouping as well. And so in terms of prediction error, what we found, we have the two regressions here, the child oral versus the maternal oral. And here we're again in orange. And so this is the median number of features selected after doing this process a few times. And so what was interesting to us is the non-robust methods, and this is something they actually had an issue with in the original paper, um, is depending on the approach you use, here the lasso produced extremely sparse solutions, having trouble selecting more than one feature, and oftentimes only selecting the intercept. So there's something going on skewing it to where nothing seems to be relevant. In contrast, all of the robust methods were producing very dense solutions with around 50 variables each, with only around 80 features that you're really looking through. So there's not much selection happening at all. And then ours, I guess, was somewhat in the middle with around 13 features. And we took a closer look at the features to say, hey, does it pass the lab test? Does it at least make sense what we're picking up? And fortunately, our selected groups of abundances and even the effect directions of those groups were pretty consistent with existing literature for the ones that weren't novel. There were a few that we didn't see anything, so it either was noise or maybe it's something new for doctors to look into. But for many of them, they were related to other types of bacterial abundances that were also associated with things like maternal weight gain. So it wasn't crazy that they might also be connected with infant weight gain or the CWG score. In terms of prediction error, I say competitive because we didn't win every time. You could see in the child oral regression case we're a little bit higher than at least the sparse LTS model. While we did end up getting lower prediction error on average than the other models under the maternal oral regression case. And so hard to say. Again, it's a difficult problem, so we don't expect the best prediction accuracy, but we know that we're at least competitive while not having to include 50 features to work with, right? So a simpler model with comparable prediction isn't a bad compromise. Finally, we wanted to actually take a look at what these outliers seem to be. Like, do they connect to anything in particular in the data set? Are we just trimming out nothing? Is there not a big difference? And so if you look at robustly standardized residuals from like an overall model on the full data, so applying the entire selection and outlier detection procedure to it, you can see here we have the child oral versus maternal oral regression residuals, and you get some extreme values occurring, which we then took a closer look at here to get an idea of if I compare my maternal oral regression residuals against my child oral standardized residuals, you can see that the most extreme scenarios were identified as outlying in both cases. So there is consistency happening among both regressions. And if you look at, again, the most extreme ones, we really dug through all the survey information, any other demographic information, just to see, is there maybe a way to explain why these kids in particular were detected as most extreme across the board? And they happen to also correspond to children whose mothers smoked while pregnant. And so there does seem to be some effect here that might be going into why they had more extreme bacterial abundances in some directions. Some of the kids had extreme changes in weight. And so it was, I guess, nice to see that it seemed to match something we can actually find in the data. And there might have been some subgroups there that we might not have been aware of otherwise. And so just to summarize, to leave room for questions, I provided a general framework for simultaneous feature selection outlier detection. We proposed mixed integer programming to do this, again, really heavily leaning on L0 constraints to induce sparsity. We have optimality guarantees as a nice benefit of using some of these solvers, along with some theoretical guarantees that go beyond just a high breakdown point, as other robust methods tend to lean on. And finally, hopefully the numerical results to everyone here also help support these theoretical guarantees. So we try to move past theory, move past simulations, and see, okay, this was actually helpful for something in a genuine case study. And then we've also have applied this now on other applications, as I mentioned, like the BE application. So with that, I want to thank you all for listening, as well as my collaborators, Luca, Francesca, and Giovanni. and the funding agencies who supported me during my PhD. And I'll stop there.
Thanks, Anna. Does anybody have any questions? You can either type them in the chat or come off mute and ask.
I'll ask a question. Yeah, I guess I have two questions maybe. But if someone like hands us a data set, is there any like heuristics we can use to select a method? Like, should we use this mixed integer programming method, or should we use LSO, or should we do both?
I always lean towards both. It also helps lend some stability to results if at least you're seeing some similarities across multiple approaches. If we want to think rules of thumb, we know that lasso pretty heavily breaks down under correlation settings in your features. So if you see a lot of block structures and things like that, lasso is likely to have a harder time with that sort of thing. I guess it is one easier one. If you have a huge data set, and I'm talking millions of features or something, then you're probably going to want to do more of a screening first to try and filter down to maybe some relevant set of features before even using a lasso or a mixed integer approach, just because it's so big that you need to help out some of these selection methods to get them going. There are a few others, but I guess those are some go-tos for me.
Okay, thanks.
I've got a question for you, Anna. In your mixed integer program, do you have any guidance on how to choose some of the hyperparameters? You had like this KP, which was your upper bound on number of features, and KN, some upper bound on number of outliers. How do I choose those parameters?
Great question. We have a whole appendix full of stuff on that part. But I would say what we ended up using were BIC, so pretty simple tuning methods often used for specifically feature selection tuning. We also did cross-validation as another approach, which is more data-driven, versus BIC relies on some asymptotics for deciding which of these Ks to use. And so in practice, again, heuristically speaking, I would say the BIC performed best, and that's why we ended up using that here. But there are a lot of different tuning approaches you could take: BIC, AIC, cross-validation. I tend to lean towards cross-validation if I'm not sure if my data is going to fit any given assumptions, just because it's strictly data-driven itself.
Thanks. Any other questions? We probably have time for one more quick one.
I've got a question. So in your mixed integer program, you have some big M values. Do you have a way of choosing like choosing the M values? Like you want to make sure they're the appropriate size so that that everything is functional.
Yeah, so there are some smaller first order methods that I believe the MIT group proposed that's supposed to be more accurate. That's in that 2016 paper I linked. But another way we did was you can start off with some of the robust estimates coming from things like the robust lasso or the elastic net. And then we like multiply them by a constant to make them bigger and then try. And then from there, you also want to do a few runs where you see how much is your solution varying if you adjusted some of those scalar bounds a bit. If it's really sensitive to that, then you might want to go bigger to make sure you're not influencing the fit. Because the M bounds, I think as you're saying, they're not supposed to really cause shrinkage or be affecting that. It's just strictly to help make sure it's going to be zero when it should be, right? But if you make them too wide, you're never going to solve your problem, I guess is the other issue. So two routes, I would say. And we tried both, the one with the MIT group, and we didn't see a huge difference. But again, this was after we had kind of checked ours a bit to make sure they were pretty stable. Yeah. Does that make sense?
Yes, thanks.